Hello everyone, I am Chiara Braconi, a clinician scientist at the University of Glasgow and a medical oncologist at the Beatson Cancer Center. I'd like to thank the AMMF for the invitation to this meeting and giving a talk about how we can optimize the use of chemotherapy in cholangiocarcinoma patients. So why talking about chemotherapy? Because at the moment, chemotherapy represents the cornerstone of treatment for cholangiocarcinoma patients. About 20% of patients currently undergo curative surgery and they will be offered chemotherapy after the operation. 60% will receive chemotherapy as part of the palliative program. And yes, there will be about 20-25% of patients who might be suitable to targeted therapies, but remember, these will be a second-line choice after chemotherapy has failed. How will this scenario change in the future? Well, we are trying to improve the data from the adjuvant treatment and assessing if chemotherapy with cisplatin and gemcitabine might be better than the tablet. We know in the palliative setting that addition of another chemotherapy drug may be useful to improve the results of the cisplatin and gemcitabine combination. And yes, probably we will be able to bring some of the targeted therapies up front, saving chemotherapy for some of our patients. And yes, we might have some more targeted therapies coming up in the future, fortunately, but these will still represent not the majority of cholangiocarcinoma patients. So at least half, if not more, of our patients will undergo chemotherapy in the future. And this is why if we improve our knowledge on chemotherapy resistance, we can achieve different goals. First of all, we could better select the patients who have a long-term benefit from chemotherapy. Think about the data from the ABC02 trials, which tell us that about 10-11% can have a long-term benefit from cisplatin and gemcitabine combination. We can spare useless toxicity to some patients, improving their quality of life. And we could come up with new chemosensitization strategies. And if we really understand the biology underpinning drug resistance, then potentially we could come up with vulnerabilities to develop novel therapeutics. So when we talk about chemoresistance in cholangiocarcinoma, we need to keep in mind that there will be some factors coming from the tumor cells themselves, and also some contribution from other cells of the tumor microenvironment that can communicate one each other and with tumor cells. So our team has had a long-term interest in understanding drug resistance in cholangiocarcinoma, and we have also liked to take two different approaches. One, looking specifically at the tumor cells, so focusing on one player, but trying to look at the dynamic conditions and the dynamic response of these cells to chemotherapy. And on the other side, taking a more holistic approach, looking at cholangiocarcinoma tissues, where despite studying static conditions, we could take into consideration multiple players. So if we start by looking at the factors intrinsic to tumor cells, one of the interests of the lab has been non-coding RNAs. This is part of our genome that does not encode for proteins, but seems to be the more abundant. And also it's the one that justifies the differences between human and other species genome. In fact, the quantity of non-coding RNAs within a genome is proportional to the complexity of the species. This really underlines the essential function that the non-coding RNA can play within the cells, either normal or tumoral. Now, there are different types of non-coding RNAs, and for the purpose of this presentation, we will focus on microRNAs, which are already known to play a role in cholangiocarcinoma development and progression. We wanted to really understand how microRNAs could drive the functional changes occurring within cholangiocarcinoma cells while they were exposed to chemotherapy. So we took a functional high-throughput screening approach where we could inhibit about 1,000 microRNA at the same time while cells were live undergoing chemotherapy stress. And cutting a long story short, we were able to identify about a handful of microRNAs that when inhibited, they could increase the sensitization of cells to chemotherapy. And combining this in vitro data with the study of human tissues, we could shortlist a candidate, MIR-1249, which not only is overexpressed in 30 to 50% of human cholangiocarcinoma tissues, but it also seems to be associated with the prognostic value. The interesting point is that we noticed that the action of the MIR-1249 inhibitor in reducing cell viability of cholangiocarcinoma cells was present only if the cells underwent treatment with chemotherapy. So no differences were observed when cells were not treated, leading us to hypothesize that this microRNA could actually drive the survival and growth of chemo-resistant cholangiocarcinoma cells. So in the hypothesis that we have a mixture of chemo-resistant red cells and chemosensitive green cells in cholangiocarcinoma tumors, then we can expect that chemotherapy determines an enrichment of the chemo-resistant cells after treatment. And mye-1249 could be involved into the expansion of these red cells. In fact, we could see that these red cells have a higher expression of mye-1249, as well associated with an expression of CD133 in their surface. When we enforce the expression of mye-1249 in cholangiocarcinoma cells, then we see that there is expansion of this subclone of CD133 positive cells, cells that also have the features of cancer stem cells. Conversely, if you disrupt the expression of this microRNA within cholangiocarcinoma cells, you can notice that the usual enrichment of CD133 positive cells that we tend to see after chemotherapy is completely abrogated, and the cells become more sensitive to chemotherapy in a dose-dependent manner. And interestingly, this effect is completely rescued by the re-overexpression of mye-1249 in these cells that have been genetically modified to lose the expression of this microRNA, making this effect really specific for the microRNA itself. And we also noticed that disruption of mye-1249 was necessary for achieving a shrinkage of the tumor identified by the line going below the zero line that we usually don't observe when we treat the mice with chemotherapy without disruption of mye-1249. So from this data, we knew that mye-1249 can mediate chemoresistance in cholangiocarcinoma. And because we also know that microRNAs can be detected in the plasma of patients, one of the things we can speculate is that assessing circulating mye-1249 in cholangiocarcinoma patients could help to select those patients who do not benefit from the treatment. So if this is something that from a clinical perspective definitely needs some validation, it's also something that gives us some insight into the biology of cholangiocarcinoma. But there are still some open questions. Does the tumor microenvironment that we talked about at the beginning contribute to these mechanisms? Does it provide a source for mye-1249? Well, Francesco Amato, who is a PhD in my team, is now trying to address this question and is setting up co-cultures between cholangiocarcinoma cells and myeloid cells. So why did he choose myeloid cells? And this is based on the data that we have achieved starting from that different approach of looking at cholangiocarcinoma tissues. In this study, for example, we have looked at resected biliary cancers, where we have been able to retrieve the tumor and the peritumoral area. We microscopically dissected the tumor from the peritumoral area, and then we ran a immunoprofiling, looking at gene expression. And we could see that there was a higher number of genes in the peritumoral area that when deregulated could actually impact on the relapse of the patient. And when we extended the cohort, we validated some interesting genes, such as CTLA4, that you all know as is a target of the current immunotherapeutic drugs used in the clinic. Now, CTLA4 is usually expressed on the Treg cells, which are immunosuppressive cells, which need to interact with other molecules like CD8 and CD86 to exert their immunosuppressive function. So when we looked at the expression of these genes, we could see that CD80 had a prognostic value, but only in the group of patients who received chemotherapy after the operation. And these data were also validated when we looked at the protein expression. Like you can see here that the expression of CD80 protein is associated with a worse outcome in patients who receive adjuvant chemotherapy, but not in those who don't. And if we want to look at this from a different perspective, we could also hypothesize that probably the patients with a tumor that have negative or mild CD8 expression are those who can benefit from adjuvant chemotherapy, conversely to those who have a strong expression. Now, this study is just hypothesis generating. The numbers are too small to really draw conclusion. And this is why we are now trying to scale it up and look at bigger numbers using the cases from the BILCAP study. So in collaboration with the group from the BILCAP, John Primrose, Bridgewater, Peter Fletcher, and also Dr. Vincent from Padova, we have stayed for CD80 the cases of the BILCAP study, and the analyses are ongoing, and we really hope to give you some updated results soon. So from this study, we learned that CD80 could potentially in the future represent a marker for selection of patients for adjuvant chemotherapy. But also from a biological perspective, we really know that CD80 is expressed mainly on macrophages. And so suggesting that there might be a role for myeloid cells in modulating drug response in cholangiocarcinoma. Now, the question is still open if understanding this is only related to the primary tumor and the microenvironment of the tumors that when they arise from the primary sites, or if this can be extended also in advanced cholangiocarcinoma where other mechanisms can take over. Recently, we've done a collaboration with the group by Jesper Andersen in Copenhagen, with Colm O'Rourke and Max Salati in Modena, and we were interested in understanding the transcriptomic of advanced intrahepatic cholangiocarcinoma patients undergoing chemotherapy. So we started from a pilot study, a pilot cohort of a small number of cholangiocarcinoma patients, but with all homogeneous characteristics. And we divided into groups according just to one feature that made these patients different, and that was survival outcome. Some patients were treated with chemotherapy and were alive after 24 months, and some patients were treated with chemotherapy, but unfortunately they did not get any benefit and they died within 6 months. We retrieved the pre-treatment liver biopsies from these patients. We also assessed them through digital pathology to make sure that the different components of the tumors were distributed across the different groups. And then the transcriptomic analysis showed that there are some genes that can differentiate these two groups. In particular, there are 500 genes that seem to make to differ between these two different types of samples. Now, the interesting point is that if you go and look deep at these genes, you realize that they do occur mainly in some cell subtypes in cholangiocarcinoma. And the cells which undergo reprogramming according to their prognosis, the prognosis of the patients, are mainly tumor-associated macrophages or tumor cells. And these are just few of the data that we gathered. And if you are interested in learning more details, you can listen from Colm O'Rourke presentation, who will present the data at this meeting. Now, we have tried to set up this score so that this could be assessed prospectively for treatment stratification, and this is something we will be interested in studying in clinical investigation, but also from a biological perspective. But this gave us an insight on how the differences between long-term survivor and rapid progressors of patients undergoing chemotherapy can actually come mainly from the interaction between epithelial cells and macrophages. So now Francesco, in collaboration with Colin Rev in my group, are really interesting to understand how these two cell types can interact together, and they are setting up co-cultures between organoids and monocytes from the patients. So Colin has had an interest over the last year to derive organoids from cholangiocarcinoma patients, and also try to understand how the organoids can help in informing drug treatment in cholangiocarcinoma patients. Now, you all know about patient-derived organoids. These are mini tumors that can be used as a functional model to resemble cholangiocarcinoma. And here you just see some of the examples that we might recapitulate. In this case, for example, on the left, we have a chemorefractory intrahepatic cholangiocarcinoma. We have ampullary lesions with different degree of malignant differentiation. And we also have, for example, chemo-naive distal cholangiocarcinoma. Not only the organoids or mini tumors can mimic what happens from a morphological perspective. You see the analogies between the organoids and the biopsy, the tumor, the source tumor, but also from a transcriptomic and a genetic perspective, the organoids can mimic what happens in the tumor itself. And the interesting point is that this molecular phenotype can be maintained over culture. So potentially we can derive these mini tumors from the patients, culture them, and then after when they are ready, we can test different drugs on these organoids. This is our experience of intrahepatic cholangiocarcinoma organoids from a patient who was chemorefractory. He had progressed, unfortunately, to oxaliplatin and fluorouracil, and you can see that also the organoids are resistant, as depicted by the green dots. But you can see that there are some other actually compounds that might be useful for this patient. Now, this is a perfect approach for a drug discovery project when we can see some potential promising drugs if we want to take them in the future. And we could test the drugs directly on the organoids, or we could modulate the organoids from a genomic perspective to really see if we wanted to understand better about a specific involvement of genes or biomarkers. But there is a step further we could think about organoids, which is a live drug screening. Now, this is when we have been able to contribute to a co-clinical trial. In this case, we're not cholangiocarcinoma patients, but it was a mixture of gastrointestinal cholangiocarcinoma, gastrointestinal cancer patients who underwent their treatment at the Royal Marsden Hospital. And in parallel, we have been able to derive mini tumor and organoid from all of these patients and treat the organoids exactly with the same treatment that the patient was receiving in clinic. So what we could see is that there was the capacity of the organoid to mimic the response that the patient had in clinic. And this is just one example. These are 3 gastric cancer patients. 2 of them turned out to be resistant to the chemotherapy paclitaxel, and 1 was sensitive to paclitaxel. Now you see that the organoids that we derived from the tumor of these patients had exactly the same trend. So the organoids from the tumor of the resistant patients did not respond in vitro, while the organoid from the sensitive patient did respond in vitro. And when this patient unfortunately progressed and we derived organoid from the growing lesion, we could see that now this organoid stopped benefiting from chemotherapy exactly as the patient had stopped benefiting from the drug. So this is what we are trying now to do in Glasgow. We are trying to combine a program of organoids derived from patients before they start the treatment and liquid biopsies to see if we can inform better a second-line treatment. And with this, I would like to conclude and to thank everyone who has participated in the study that I presented today and all the supporters that are contributing to the activity of the lab. Thank you very much.
Good afternoon and welcome to the penultimate Q&A session of the virtual European Cholangiocarcinoma Conference. Helen and I are delighted to be joined by Chiara Braconi, who is here to respond to your questions following her recent session on optimizing chemotherapy activity with the use of novel biomarkers. So, to start off the session, I'm going to hand over to Helen, who will start by asking a question we received a moment ago.
Thank you, Gary. Chiara, wonderful presentation. Thank you so much. And it's heartening to see the amount of work that's going on, and your presentation belies how much hard work has gone in behind that. So first question is relatively general, but I think it's a really important one, is how do you see the role of liquid biopsy in cholangiocarcinoma?
Well, definitely I see there will be a role for liquid biopsy. Can you hear me now? For liquid biopsies in cholangiocarcinoma. And I think that we probably need to think about liquid biopsy in a different way compared to some other tumors. In other tumor types like colorectal cancer, for example, we are also using liquid biopsy to monitor the efficacy of a treatment. And I'm thinking about cetuximab treatment, for example, and see how mutations can arise that can justify the resistance. In cholangiocarcinoma, I see a role for liquid biopsy mainly in assessing which are potential molecular alterations that could be targeted for treatment. We have listed in yesterday presentations as well as today that there are a lot of different markers that could be used for therapeutic development. And we have seen that actually cholangiocarcinoma is quite rich in these molecular markers. And sometimes it's difficult to define in a patient which is the driver of the tumor, because as you all know and has been discussed, the achieving tissue in some cholangiocarcinoma patients is quite difficult, and the patients are aware themselves, especially if you have distal or periodic cholangiocarcinoma, might be difficult. So liquid biopsy may be a good surrogate to look at the DNA of cancer cells and how they can be targeted. So definitely there is a role there that can be used.
Well, thank you. That's a really interesting and full answer. And I'm going to go straight on, if I may, Gary, with another question, which really sort of leads on from that, I think. And forgive me if you've actually answered it in what you've just said, but could liquid biopsy be used in clinical practice to detect their MRI 1249 and help identify those patients who may not respond to chemotherapy?
Well, this is definitely not a test that we can run now in clinical practice. This is still, a research field. We, from the data that we have seen, there is a potential that this microRNA could be detected in the plasma of patients. So with a simple blood test, we could identify which patients undergo a better response to chemotherapy. So what we are planning to do now is to put together a lot of different patients, potentially in prospective clinical trials that of patients undergoing chemotherapy would even be better, so that we can really understand which is the role of NIR1249 as a biomarker of response to treatment.
Okay, that's great. Gary, what's...
Excellent. We've just had a question come in through the live chat from Dr Ed Jarman, and it's great talk, Chiara. What cell types are represented in patient-derived organoids? And are innate immune cells such as macrophages present, for example? Is recapitulating immune roles in these models an important next step? There's about 3 questions in there, Chiara. I'm sorry about that.
We will also have the chance to answer some of the questions in the next workshop. So organoids is definitely a promising tool that is very dear to me, but is not flawless. So probably what Ed is pointing out is one of the major limitations of organoids, which is the absence of all the microenvironmental cells. Now we are trying to address these issues by creating organoid chips where we can put the organoids coming from the tumor with other cell types, and these can be immune cells or can be CAF or endothelial cells. And what we have seen when we grow organoids is that probably over the time there are only epithelial cells that you can collect and you can study. But my idea and my my thought is that these cells are imprinted from their being in the microenvironment of the tumor. So they actually, this gives them the possibility to recreate this 3D structure in matrigel that resemble better the morphology of the primary tumor. But then we won't get, they will not survive the immune cells that will not be there.
Wonderful, thank you. I'm going to go to another question that's immediately just come in from one of our previous panelists on the Q&A, Dr Toby Fessy. Great talk, Chiara. Do all the patient-derived CCA organoids grow in a common growth media, or do they need to try several per sample?
Organoids, I think that we still don't have a lot of expertise generally worldwide in cholangiocarcinoma organoids to really understand which is the efficiency rate in biliary cancers. Compared to other tumor types, like for example colorectal cancer, we have seen that these are more challenging. They tend to be more similar to pancreatic cancer, for example. In terms of growth factors, we didn't have necessarily to change the media for growing organoids, but what we had to do is be very careful in the way we were splitting the organoids, and use different technologies to do not repeat a single cell dissociation, because that could have affected the possibility for them to continue in the culture.
Thank you, Chiara.
Coming in from, well, they're pouring in at the moment, but from Sheila Jayaraman. And I'd like to put this one to Chiara. Again, Sheila has very much enjoyed your talk. And she says, does the microRNA that's associated with resistance related to DNA damage response?
We don't know, because we haven't actually tested this specific question in our project. Not for these specific microRNAs. We were more interested in understanding the potential of this microRNA to let CD133 positive cells grow. Because those cells, we were showing that they have kind of cancer stem cell feature. So potentially those are the ones that really can stay there when a patient is given chemotherapy, and those are the ones responsible for a tumor to grow even after a potential response. But we know that microRNAs can do that. And for example, we are running a parallel study now in pancreatic cancer, and we see that microRNAs can definitely change the way cells respond to DNA damage.
I'm sure that Sheila will probably come back to you on that if she'd like to take that further, because I know they're doing some really good work up there in Nottingham at the moment. So she'll probably want to contact you offline, I'm sure, if you're not already in touch. I'll move on again, if you don't mind, Gary. We've got a question coming in from Luke Bolter. I don't think we've done this one yet. How do you envisage modulating macrophages clinically, preventing recruitment into the tumour, or through limiting their differentiation? And do you have a preference?
Well, I think that this is a very interesting and focused question, as this is actually where I think we have the potential to impact on chemoresistance and in general on therapeutics for cholangiocarcinoma patients. From the data we have from other tumor types, we know that if we deplete macrophages and we are not able to deplete the tumor-associated macrophages, the toxicity for the patient can be too high. From the data that we have done in collaboration with Andersen's group, we have seen that there is a rewiring of macrophages, transcriptomic program between responders and non-responders. So try to change that programming probably would be a good choice. I guess that what we lack now is the knowledge of which are really the key components of genes within the macrophages or monocytes that can predispose cancer cells to chemotherapy. So I think that we really need to dig out in that sense before we can propose a treatment.
Great, thank you very much for that. And I'm sure, again, if that's opened up more questions for Luke, he will be back to you on that one. Have no fear. I think that may be all we have time for, Gary. Is that right?
It is. We are perfectly timed on that session. I'm sure there are other questions.
Yeah.
To be answered, but as you mentioned, there's another Q&A with Luke and Chiara after this, so maybe some of those questions will be responded to in that session as well.
If we let Chiara can look at the questions, and if they're pertinent to the next session, do by all means bring them in, and I'm sure Luke will have an eye on those and he'll be interested. But huge thanks, Chiara. That was really fascinating, and I always enjoy listening to your work and how much you're pushing the boundaries for us. So that's great. Thank you very much.
Thank you, Ellen, and thank you everyone and Ellen for inviting me and being here today. It's really a pleasure.
Thank you.